We're going to begin a series of podcasts on the lymphoid system. In this series, we'll give you an overview of the immune system. We will talk about the lymphatic vascular system, and then we will in turn talk about each of the major lymphoid organs. So the first podcast will be an overview of the immune system, and I want to stress this is just an overview because you will have an entire course next year in your medical school training on immunology. Now, the learning objectives for this entire series of podcasts, you should be able to describe the major cell biological aspects of the immune system, and you should be able to recognize the major lymphoid organs at the microscopic level and be able to explain their histological organization in relation to their immune function. This is a table that we'll come back to at the very end of the podcast series that will help you consolidate information on the lymphoid organs. So what are we going to do? This first podcast is going to be an overview of the adaptive immune system. We will talk a little bit about the molecules and cells involved in the adaptive immune system. And then subsequent podcasts, we will look at the lymphatic vascular system and the histology of the various lymphoid organs. If we start off with just an overview of the immune system, we can say the immune system provides the second and third line of defense that the body has against invading pathogens. Now you might imagine that the first line of defense is simply the epithelium that covers every surface of the body. So you could say the skin, in a sense, or the epithelium lining the internal organs is the first line of defense against invading pathogens. Beyond the first line of defense, we have the second and third lines of defense. We can talk about the innate immune system, and the innate immune system consists of macromolecules. You may have heard of some of these, like the complement system, various secretory substances and enzymes, as well as cells, and these cells might be macrophages, neutrophils, mast cells, and other natural killer type cells. So these are all part of the innate immune system. The innate immune system responds fairly rapidly, but it is not specific. There's no memory involved in the innate immune system. It's a more primitive immune system from an evolutionary perspective. And then a more sophisticated line of defense comes from the adaptive immune system. That's a complex system. It's relatively slow in its response. It's highly specific. The adaptive immune system does involve macromolecules, that is antibodies, and it certainly involves cells, the lymphocytes and antigen-presenting cells. And we can describe a primary immune response and a secondary immune response as part of the adaptive immune system. This is a fairly complicated slide. It's just highlighting various formed blood cell elements and the cells in the immune system. And I'm not going into this slide in much detail right now, but I think people are aware that there is a hemopoietic stem cell that, under the influence of various transcription factors and signaling molecules, will differentiate into a common myeloid stem cell and a common lymphoid stem cell. The myeloid stem cell gives rise to the blood cells like the erythrocytes, platelets, monocytes, eosinophils, basophils, neutrophils, and the lymphoid precursor gives rise to B lymphocytes and T lymphocytes. Now, I think you already appreciate that the blood cells develop in the bone marrow, and lymphocytes develop in the bone marrow and in various peripheral lymphoid organs. And we'll be talking about that later in the course, and we'll talk about the development of the lymphoid organs later in our podcast series. This is just a broad overview of hematopoiesis in embryos and in the fetus. And it's just showing when the various cell lineages begin to form. We have on the x-axis here just months of gestation with birth indicated here. And from the point of view of the immune system, I just want to highlight for you that the earliest lymphoblasts begin to develop around 4 months of fetal life. And they're developing pretty prominently in the fetal liver, a little bit less so in the spleen. Right around 4.5, 5 months, the bone marrow begins to be very active in the development of all of the lymphocytes and blood cells. And by about the 7th month, the bone marrow is primarily involved in blood cell and lymphocyte development. And then at birth, the primary and secondary lymphoid organs also will play a significant role in the development of lymphocytes. Some characteristics of the adaptive immune system. First of all, we can talk about its specificity and the fact that it's self-limited. And by that we mean that specific domains of antigens can be recognized by antibodies or by lymphocytes. And self-limited in the sense that when the antigen is neutralized, an immune response ends. With respect to diversity, and this one is a little bit tricky to sometimes wrap our minds around, but basically molecular mechanisms are in place that ensure that the body is pre-programmed to react to just about any antigen it would ever encounter. With respect to memory, we know that mitosis leads to clonal expansion of effector cells and to reserve or memory cells. That means that on a subsequent exposure to the same antigen, the immune response is much faster and more vigorous. And finally, the adaptive immune system has the ability to recognize self from not self. It is very critical that as the immune system is developing, tolerance is achieved so that there's selection against immune cells that would react to self-antigens. You know that sometimes this goes wrong when we would have autoimmune disorders. Can say there are two arms in the adaptive immune response. There's a humoral response, which is an antibody response. That involves B cells or B lymphocytes. B lymphocytes differentiate into plasma cells or to B memory cells. We'll come to appreciate that B cells respond directly to an antigen, and in humans, we say that B cells are derived from bone marrow. It turns out that B cells are also derived from gut-associated lymphatic tissue. And a little bit of historic perspective. B cells were first identified in the middle part of the 20th century in an organ in birds called the bursa. Experiments showed that if the bursa was removed from birds, the birds would have no humoral immune response. And so the B cells were given the name B cell to signify they came from the bursa in birds. And then the second arm to the adaptive immune system is the cell-mediated response. And the cell-mediated response involves T cells or T lymphocytes, and there are a variety of T lymphocytes. So, for example, there are two classes of T helper cells. There are T memory cells. There are cytotoxic T cells and regulatory T cells. Now, to contrast the ability of T cells to respond to antigen with respect to B cells and their response to antigen, T cells, unlike B cells, cannot respond directly to antigen. They have to have the antigen presented to them by other cells in the body. We call these cells antigen presenting cells. And antigen presenting cells could potentially be any cell in the body, or they can also be a specialized type of cell, and we'll talk about that a little bit later. T cells get their name because to gain immunologic competence, the cells must mature in the thymus. The stem cells for T cells originate in the bone marrow, but they don't gain immunologic competence unless they mature in the thymus. The basis of a B and T lymphocyte response in an immune reaction will initiate mitosis for clonal expansion, and this clonal expansion is critical in providing immunologic memory. Some basic terms. I think everyone is familiar with these, but just so that we're all on the same page. An antigen can also be described as an immunogen. It's a foreign structure capable of eliciting an immune response. An epitope is an antigenic determinant or a region within an antigen that reacts with either an antibody or with a T cell receptor. And as we've suggested, the body is pre-programmed during development to react to almost all possible antigens. And we know this occurs because there's rearrangements in the gene sequences that code for the various immunoglobulins, and this post-translational modification of the proteins, either for the immunoglobulin proteins or for the T cell receptors. T and B cells, as we've indicated, when they respond, they undergo a clonal selection and a clonal expansion, and it would be those cells that divide and expand that would not react to self. On the other hand, there's a process called clonal deletion or clonal anergy. Immunocytes, whether they're T cells or B cells, that respond to self usually will get eliminated. If you don't have this elimination, that leads to autoimmune disorders. So this is what we mean by self versus not self. The body will respond to antigens that are not of itself, as opposed to it should not respond to antigens that are self-antigens. The immunoglobulins, you probably talked a little bit about these in your biochemistry classes. We have 5 immunoglobulin isotypes: IgM, IgD, IgG, IgA, and IgE. IgM is a pentameric immunoglobulin, and it is the first antibody that's made by B cells. IgD is expressed on the surface of B cells, and it's expressed as B cells mature to plasma cells. You may remember that IgG is the major circulating immunoglobulin in the blood. It also crosses the placenta. IgA is the major secretory antibody, and it's usually a dimeric immunoglobulin. And then finally, IgE is on the surface of mast cells and basophils, and it's involved in allergic reactions as these cells degranulate. Just something to remind you of if you've not seen it before, and if you have seen it before, this is just to remind you, there is antibody class switching as B cells develop. So what happens, you've got a stem cell, and as the stem cell is dividing and differentiating, it's going to be a naive B cell. It will begin to express some immunoglobulin chains on its cell surface, and even as it continues to develop, we call it a naive or immature B cell, it will express IgM on its cell surface. As the cell matures, it'll switch production from IgM onto IgD. Membrane-bound IgM is the first antibody that is synthesized by B cells. As we said, as the B cells mature, they will begin to produce IgD. And they will be producing IgD as they're circulating through peripheral lymphoid organs. Just to remind you of the basic structure of antibodies, you would have had this in biochemistry. Immunoglobulins have 2 heavy chains shown like this, and 2 light chains shown like this. And there are antigen binding sites at the ends of each of the light chains, and then there's a relatively constant region near the C-terminal end of the heavy chains. The diagram on the right is just showing IgA dimer, and it's just highlighting the role of J protein here to hold the dimer together, and it's also highlighting the role of the secretory component on the IgA dimer. This is a schematic representation of a gland, like the mammary gland, for example. Here are the epithelial cells of the glandular epithelium. Here's the lumen of the gland. In the connective tissue here might be plasma cells that are secreting IgA dimer. The basal end of this glandular epithelium, there are receptors for this IgA dimer, and these receptors will bind this dimer, and through receptor-mediated endocytosis will endocytose this immunoglobulin dimer, and then by transcytosis will transport this immunoglobulin dimer in a vesicle from the basal surface of the epithelial cells to the apical surface, where the immunoglobulin will be exocytosed into the glandular lumen, into the secretory space. And it will be exocytosed with part of this receptor molecule still attached. That's the secretory component. Now let me look quickly at the cells of the immune system. We know that there are B lymphocytes, and B lymphocytes express on their surface CD19 plus other CD molecules. B cells differentiate into plasma cells and B memory cells. Now if we say CD molecules, we're talking about cell surface receptors that are broadly given the name cluster of differentiation molecules. These essentially are molecules that can be used to identify different types of cells, and so B lymphocytes are going to express early in their development the molecule CD19 on their cell surface, so we can say they're CD19 positive. T lymphocytes will be CD3 positive, as well as expressing other CD markers on their cell surface, and T lymphocytes will differentiate into a variety of cells, including T memory cells. So for example, if we talk about T1 helper cells, we would say they express CD3, CD4, and CD28 on their cell surface. If we talk about T2 helper cells, we would say they express CD3, CD4, and CD40 on their cell surface. We know that T1 helper cells stimulate macrophages, and T2 helper cells stimulate B cells to produce antibody. HIV virus infects T helper cells. It binds onto cells basically that express CD4 on the cell surface. And an individual gets full-blown AIDS when cytotoxic T cells destroy infected helper T cells. What happens is you're wiping out cell-mediated immunity. Now, cytotoxic T cells express CD3 and CD8 on their cell surfaces. Cytotoxic T cells kill tumor cells and virally transformed cells. So there is what happens with full-blown AIDS. The cytotoxic T cells attack and destroy helper T cells that are infected with the HIV virus. And then finally, there's a class of T cells that are called regulatory T cells. These cells express CD4 and CD25 on their cell surface. Regulatory T cells have the ability to modulate the response of other T cells. This table is way more complicated than I expect you to be involved with for our course. It just shows you some of the CD markers that we know of. There are many, many pages worth of tables of the CD markers, and you'll encounter these as you go along in your clinical training. In addition, there are other cells in the immune system. Antigen-presenting cells interact with T cells. Remember, T cells themselves don't respond directly to an antigen. They have to have the foreign antigen presented to them, and this is the role of the antigen-presenting cells. There can be many different types of antigen-presenting cells. So, for example, there are epithelial reticular cells in the thymus. These are cells that are derived from epithelial cells. There are dendritic cells that are in lymph nodes and bone marrow. These are tissue-specific cells that are derived from multiple sources. There are Langerhans cells that are in the skin and connective tissue. These are cells that are derived from monocytes. There are M cells in the GI system, and B lymphocytes can also be antigen-presenting cells. Essentially, antigen-presenting cells process and display antigens on their cell surface. They're going to process and display either endogenous proteins to cytotoxic T cells by virtue of interacting with what are called MHC1 molecules, or they're going to process and display exogenous proteins to helper T cells by interacting with MHC2 molecules. MHC1 and MHC2 molecules are broadly classified as major histocompatibility complex molecules, and we'll talk a little bit about MHC1 and MHC2 molecules in just a couple of minutes. Now, there are also natural killer cells. These are cells that are part of the innate immune system. They are MHC independent, and they attack tumor cells and virally infected cells. Natural killer cells apparently express CD94 on their cell surface. That's not something I'm going to expect you to remember. Exactly what are MHC molecules? Well, they are glycoproteins that are found on the surfaces of all nucleated cells in the body. And if we look at the panel on the left side first, MHC1 molecules are for normal or abnormal host proteins, and these would be proteins that are considered endogenous proteins, that is, proteins that are found in the cell. And what happens, these endogenous proteins as antigens will be presented to cytotoxic T cells. So any cell in the body has the potential to express MHC1 molecules on the cell surface. And what happens here is an endogenous protein that's in the body. Well, we know that proteins get broken down by machineries in the cell. For example, proteasomes break down proteins, and portions of these proteins will get into the Golgi apparatus, and you might imagine that MHC1 molecules are molecules found on the cell surface. So this is an example of a cell surface MHC1 molecule. Well, you already know that a protein found on the cell surface is synthesized on rough ER. It goes through the Golgi where it's processed and packaged into vesicles to fuse with the plasma membrane, and the molecule will be inserted into the plasma membrane. Well, some of these broken down components will get attached in the Golgi to MHC1 molecules. So as those MHC1 molecules travel through the Golgi, through secretory vesicles, and through exocytosis get inserted into the plasma membrane, these MHC1 molecules will display on the cell surface epitopes of the endogenous proteins. And these epitopes will be presented to cytotoxic T cells, and that's part of how the cytotoxic T cells are involved in immunologic surveillance. On the other hand, we talk about a specialized type of cell called an antigen-presenting cell, which expresses on its surface MHC2 MHC2 molecules, and these are for foreign proteins. We call these exogenous proteins. So in one sense, the process is very similar. The MHC2 molecules synthesized on rough air go through the Golgi, get packaged into secretory vesicles to be exocytosed and displayed on the cell surface. At the same time, the specialized antigen-presenting cell may be endocytosing a foreign body, like a bacteria or some foreign substance. This foreign body is broken down in vesicles. Some of the components fuse via endosomes with the MHC2 molecule as it is going through the biosynthetic exocytic process. These MHC2 molecules bind onto the exogenous protein epitope, and this gets bound onto the MHC2 molecule to be expressed on the cell surface. So to summarize this slide, specialized antigen-presenting cells recognize exogenous proteins interacting with MHC2 molecules, whereas any cell in the body, by virtue of expressing MHC1 molecule, can present endogenous proteins to immune surveillance cells. So the endogenous proteins are presented to cytotoxic T cells. The exogenous foreign proteins are presented to various helper T cells. This is a very complicated slide. I'll try to simplify it as we go through it. We're going to look at the role of MHC1 or MHC2 molecules in how they react with endogenous or exogenous antigens. We can talk first about B cells, which is just this little shaded part of the diagram. As you know, B cells interact directly with antigen. When they interact with antigen, and that's shown here, the B cell interacting with an antigen, the B cell undergoes mitosis. It undergoes a clonal expansion. It will differentiate into plasma cells, and the plasma cells will produce lots of antibody to the particular antigen, and the B cell will also differentiate into a B memory cell, so that if the body encounters this antigen another time, the immune response will be faster and more vigorous. So this is B cells interacting with a foreign substance to produce antibody. That would be the humoral immune response. On the other hand, the T cells have to have the antigen presented to them. When the antigen is presented, they will undergo mitosis and develop into various types of T cells. They're either cytotoxic T cells or T1 or T2 helper cells. And so here is an example of what happens. An antigen presenting cell recognizing these foreign antigens. These antigens are going to bind onto MHC2 molecules, and in this case the antigen presenting cell will present the foreign antigen to T helper cells. If it's presented to a T2 helper cell, the T2 helper cell will secrete cytokines to stimulate B cells to make antibodies to the original foreign antigen that is presented by the antigen presenting cell to the T helper cell. On the other hand, it may present this antigen to a T1 helper cell, which will stimulate macrophages or will stimulate cytotoxic T cells. So this would be reacting to an exogenous foreign protein. What about a virally infected cell? This would be a situation where the viral particles are in the cell, and in this case the viral particle proteins are actually like an endogenous protein. This will be presented on MHC1 molecules to cytotoxic T cells. The cytotoxic T cells will bind onto this virally infected cell and will destroy the virally infected cell. As the cytotoxic T cells are differentiating, they're making many more cytotoxic T cells as well as cytotoxic memory cells. Here, the T1 helper cell will stimulate the cytotoxic cells. Here, the T2 helper cell will stimulate the B cells. So this side of the diagram is the cell-mediated response. Some clinical correlations that we would like you to keep in mind. If you talk about antigenicity, the greater a molecule's complexity, the greater is the potential for antigenicity. On the other hand, very large man-made polymers are relatively simple, thus they have minimal antigenicity. And man-made molecules can be used in a lot of implants, and implants generally don't cause an antigenic response or an immune response. Autoimmune diseases, as we've hinted, where you lose a self-tolerance. The body loses the ability to not respond to self molecules and instead mounts an immune response. So, for example, Graves' disease, receptors for thyroid stimulating hormone can be perceived as foreign, and thus the body will make antibodies that will cause the thyroid to make excess thyroid hormone. Another autoimmune type disease where you lose self-tolerance, type 1 diabetes. Surface molecules on B cells of the pancreas may be recognized as foreign, and the body will mount an immune response and will destroy those insulin-producing cells. As we've already mentioned, HIV/AIDS, the AIDS virus binds to cells that express CD4 on their cell surface, so the virus infects T1 helper cells, and patients will succumb to secondary infections because macrophages are less functional. T1 helper cells happen to stimulate macrophages. Another type of clinical correlation, for example, DiGeorge syndrome. You have a congenital failure of the thymus to develop. Patients that have DiGeorge syndrome cannot produce T cells. They thence lack all cell-mediated immunity, and they're going to die at a relatively early age because of lots of infections. Lymph nodes. In the presence of antigens or bacteria, lymphocytes rapidly proliferate, and the lymph node can increase in size. It may become hard and palpable on a physical exam. Because lymph nodes are located along the paths of lymphatic vessels, lymph fluid can flow from one node to another, and thus making it relatively easy for an infection or for malignant cells to spread throughout the body, as they will spread through a chain of lymph nodes. Spleen, as you know, is the lymphoid organ. Major trauma in the upper left abdominal quadrant can rupture the spleen, can cause severe bleeding in this organ, and we'll talk about why this is occurring in a later podcast. There's a relatively unusual circulation in the spleen. Traumatic injury of the spleen can lead to a lot of blood loss, but because of this unusual circulation, diseases like sickle cell anemia can cause problems. And oftentimes, if you have to remove a patient's spleen, they don't fare well because you're impairing long-term immunologic function.